So we'll move on. We have our last presentation before the coffee break. It's a great pleasure to introduce Professor Vladimir Katanaev from University of Geneva. He's an expert of this disease, has been working on this for a long time now, and we've heard about neomorphic mutations, so we're looking forward to hear a bit more about this. Thank you. Look forward to your talk.
It's a great pleasure to be here. It's amazing to see how the field has been moving forward, and really impressive. I wish my talk were after the coffee break, but anyway, you will hear to what I have to say. So this first slide shows like the different levels of analysis we try to do in the lab, starting from the patients, moving to the biochemical and structural atomistic level of investigation of what happens with the mutations, building screening platforms based on this understanding, trying to identify some drug candidates, validating them in the animal models, and then going back to the patients. I will be briefly jumping through several projects which we have done or are doing. So for example, one of the key things we wanted to analyze some years ago is what happens to the mutant protein at the biochemical level. You know that the G protein is there to bind guanine nucleotides, so to bind GTP, and then to hydrolyze it to go back to the inactive GDP bound state. So we want to see whether the mutants, they are deficient in these activities. And in fact, looking at first just these three, which are among the most frequent mutations, we find that the kinetics of uptaking the GTP is dramatically sped up, while the kinetics of hydrolysis of the GTP is essentially gone. So these mutants, they are biochemically constitutively active. And actually, now we know that this is true not just for these three, but for the vast majority of the mutants we have studied so far. And I'm inviting you to go to the poster of Ionica to see more details. We also wanted to know what happens at the cellular level. So how would the mutant proteins interact with their partners? So one of the partners which we routinely analyzed is called RGS19. And normally it should bind to the activated GTP-loaded form of the protein. And what we find is that actually the mutant shown here, G203R, although biochemically it is constitutively GTP bound, it binds this partner much worse than the wild type does. And as a control, positive control, you see the non-pathogenic mutant Q205L, which is never found in a patient, but this is a classical constitutively activated form. And this is true for other mutants, and actually for the broad panel which we have analyzed recently, the uniform feature is that the mutants lose the ability to bind to RGS19. So what is the explanation why the mutants being constitutively GTP bound are unable to bind to the partner which should recognize this form? The explanation which we provide is that the mutants actually, despite binding GTP, they fail to adopt the proper GTP activated conformation. And that's why they, despite being GTP bound, they cannot recognize their partners. We have been active in modeling the disease in animal models, mice and flies. So for mice, we have made an effort to model G203R and C215Y. So G203R was our first trial, and unfortunately, we realized that the mutants are neonatal lethal. So the mice don't survive. They are born and they die. The champion lived for 2 weeks, the runner-up lived for 4 days, and all the others are immediately dead. So with this fact, we wanted to model a disease mutation which is mild. We want to go to the other extreme. So we modeled C215Y, which was discussed earlier today, and these mice are viable and fertile, which allowed us to characterize them at the level of their epilepsy, which they do not have, as the patients do not have, and at the level of the motor activity. In multiple assays, we see that these mice are hyperkinetic, hypermotile. And we have also analyzed the brains of the C215Y mice and also of a few newborns of G203R, which were basically dead immediately, so we could still preserve their brains. And we found that there are some developmental defects. So TBR2, for example, is decreased in the levels. TBR2 positive neurons are decreased in the cortical sections, which permitted us to conclude that GNAO1 disease is a neurodevelopmental disorder. Flies, so we have seen C. elegans, which is a great model system, and of course flies is also a great model system, and G-alpha-O is extremely conserved between flies and humans, to the extent that we could successfully humanize the Drosophila G-alpha-O by introducing the human sequence by CRISPR-Cas instead of the Drosophila gene, while maintaining all the non-coding parts of the Drosophila gene. So if you remove G-alpha-O in Drosophila, this is embryonic lethal. But if you put the human gene instead, the flies are viable, fertile, they live as a colony, their duration of life is normal, their motility is normal, their learning and memory is normal. So the gene is perfectly replaceable. And this for us was the proof that flies are a valid host to model this disease. So yes, and please go to the poster of Mikhail to see more details. And so we introduced by CRISPR-Cas the G203R mutation and a bunch of others. And one phenotype which we saw is the strong motor dysfunction. So this is the simple assay called negative G-ataxis assay. So the flies have this instinct of crawling up, and you can measure the speed of that. And we see that the flies which carry one mutant allele, their movement is decreased about twofold. There is also defects in the duration of life, and there is some neurodegeneration. One question which we have been addressing is, what is the genetic nature of the mutations? Is it gain of function? And the argument in favor of this is that we see, for example, that the mutants are constitutively GTP bound, so which suggests that they are gain of function. But there are also other observations which may suggest that the mutants are loss of function, either null or hypomorph. And you know many examples from your laboratories, and we have found some observations which are published. Then you can also argue that the mutants are dominant negative. For example, you see that if you remove one copy, at least in mice and in flies, there are no phenotypes. But if you put one mutant allele over the wild type, suddenly you see the phenotypes, which suggest that this is a dominant negative mutation. Basically, how come that a mutation shares the features of a gain of function, loss of function, and dominant negative? The answer which I propose is that the mutations are neomorphs. So the neomorph concept was introduced by Hermann Müller, who got the Nobel Prize for his classical genetics works in Drosophila. And he introduced this concept of morphs, which we now call gain of function, or loss of function, or dominant negative. He called it hypermorph, antimorph, and others. Basically, I probably will jump through this. The point of the neomorph is that the neomorph is a mutation which produces a protein which has a novel function. Novel to the extent that it doesn't exist for the wild-type allele. And there are very important consequences of a mutation being a neomorph. And these consequences are schematically represented here. Specifically, if you're dealing with a gain-of-function mutation, you can suppress your phenotype by removing one gene copy of your wild type, or by pharmacologically suppressing the wild type. If your mutation is a dominant negative, you can compensate for it by producing one extra genetic copy of the wild type allele, or by pharmacologically enhancing the function of the wild type allele. In contrast, if your mutation is a neomorph, whatever you do to the wild type allele, it doesn't help. So this understanding, or this model of the neomorph, has two types of implications. One for the molecular mechanisms of this neomorphic activity, and the other for the drug discovery efforts. So for the underlying mechanisms of this neomorphic activity, we think we have discovered what is the underlying mechanism. And this is the interactions, the neomorphic interactions the mutants gain with the partners called RIC8, RIC8A and RIC8B. I invite you to go to the poster by Gonzalo, where all these details and many more are presented. Here I just want to briefly state that the mutants universally gain the massive interaction with RIC8A. And what is even more surprising, many of them, but not all of them, gain the interaction with RIC8B. RIC8A and RIC8B are like chaperone proteins which are responsible to help fold different G-alpha subunits. RIC8A is there for G-alpha O and a bunch of others, but RIC8B is a totally foreign protein for G-alpha O. So here, this is the classical neomorphic biochemical interaction which is gained. And what is even more exciting is that we see a great correlation between the clinical score, the way we as biologists could define it, and for us, the best clinical score was the onset of the disease. So the onset of the disease correlates with the strength of interactions with RIC8B. So this is the best correlation we have ever found. And what we claim is that this interaction is the biomarker for the strength of the disease. So you can come to me with your new mutations you find in your patients. We will measure how strongly they interact with RIC8B, and we will tell you, well, we will predict what is the clinical severity of the disease. There is one more thing I want to say. So there is this distinction between the DE17 and NEDIM, the two categories which actually overlap. So one thing which you will see on the poster is that the mutations which lead to epileptic phenotypes in patients, they lose or decrease plasma membrane localization. The mutations which lead to movement disorder-only phenotypes, they do not decrease plasma membrane localization. This is another major factor which we found, predictive if you wish. Implications for drug discovery. So if the disease is due to the neomorphic mutation, you cannot correct it by playing with the wild-type allele, genetically or pharmacologically. You need to find the way to affect the neomorphic allele, either genetically or pharmacologically. And we are, for pharmacology, although I am very much in favor of trying the genetic approaches too. So we have built a neomorphic assay to screen for drug candidates which would correct the deficient activity of a mutant protein. Here we were looking for the assay which would identify molecules which can bring back the GTPase activity to the mutant. So I told you that the mutants lose the ability to hydrolyze GTP, and we wanted to find a molecule which would correct this loss of the GTPase activity. And we screened 2,700 FDA-approved drugs, and we found one which can really restore this activity. This drug is shown here. It's called zinc pyrithione. You cannot dream of using zinc pyrithione to give it to patients. It's actually a topical drug for anti-dandruff treatments. But we found that the active component of zinc pyrithione is not pyrithione, but zinc. So we find the same recovery of the GTPase activity by zinc ions, zinc chloride or whatever else. So zinc restores the GTPase activity of the mutant proteins. At the same time, it does not act on the wild-type protein, so it really acts on this neomorphic feature of the mutant without affecting the wild-type, which makes it even more exciting as a drug candidate. Zinc also restores the cellular interactions of the mutants, so it's marvelous. We have done, so this work is published, but since then we have done much more biochemical studies, and actually we know that now all the pathogenic G-alpha O mutants can be grouped into three categories in terms of how they respond to zinc. The first category are the mutants which, unfortunately, do not respond to zinc at all. So here I'm showing you this Bruno mutation, the mutation in the intronic region, which we showed leads to the insertion of 2 extra amino acids in frame. And this mutant is totally insensitive to zinc. And there are some others, including them C215Y, bear in mind because this will be important later. These mutants do not respond in any regard to zinc. The second category are the mutants whose GTPase activity is restored. So the three I showed you before, K46R and probably some others. And then we discovered that there is a third category, which was really puzzling first, but then it appeared that actually we find this puzzling reaction to zinc in a multitude of mutants. So this reaction is that the mutant protein when you add zinc to it, it starts to lose its GTP. So I don't have time to go into much detail here, but the point is that the rate of GTP uptake in the mutant, which is increased, is still the same. It is not corrected, but this GTP becomes losable. The mutant loses it, and this is measured, for example, by the KD, and the KD is a proportion of K off to K on. We know the K on doesn't change, but the K off, the kinetic constant of loss of the GTP is increased by multiple folds in these mutants by zinc. So zinc salts have been approved for a multitude of disorders, neurological disorders, or for example, you may have heard of the Wilson disease, which is treated by dietary zinc supplementation. So you can buy dietary zinc in the pharmacy, of course, in reduced doses. The question is, can zinc be useful for the patients? Before answering the question, we wanted to test it in our model. So it works in vitro, in biochemical settings, in cells. Well, it also works in flies. So here in these tubes are mutant lines, the same mutant line, one raised on the control food and the other on the food supplemented with zinc chloride. And the curves and the video show you that the speed of this, in this experiment, the speed of the motility is restored to the wild-type levels almost perfectly in the line which has been raised on zinc supplementation. The duration of life is also partially restored by zinc supplementation. So, of course, we can suggest that it's time to go into patients because this is an approved drug. But of course, we want to do as many additional studies as possible. For example, the question is, can we do the tox analysis in mice? And dietary zinc has been tested for the toxicity effect in mice, but in adult mice. And we're dealing with the pediatric patients, so the question is how zinc will affect the neonatal and then developing mice. And this is a preliminary toxicity study which we performed, where we add zinc to the drinking water. And the curves show you how the mice gain weight. These are wild-type mice. And you see that they gain weight essentially normally. And the concentrations of the zinc we give, it's not very straightforward to calculate it into the human dose. But actually, the lowest which we gave here is like 30 times more than the dose which is prescribed to Wilson disease patients. We have also collected organs for some detailed anatomical histological analysis, which is not yet done, but grossly, we don't see any defects in these mice with this enormous dose. And we did some other studies on the only mouse model of GNAO encephalopathy, which we have in the lab, C215Y. I told you, C215Y is not responsive to zinc biochemically. But we still thought, well, maybe this is a proxy to a patient, because when we talk about clinical trials, the first aim, one of the aims of the first clinical trials, we hope, is the safety, and the safety on mutation-carrying patients. So we thought maybe it would still be useful to do some analysis here. So we tested zinc on the C215Y mice, and in the open field where the mutants have hyperactivity, upon zinc supplementation, there is the same hyperactivity, but at least we do not see any worsening. And we did some additional studies in the assays where the mutants actually do not show any differences from the wild type. So for example, the rotarot test, which measures how stable the mouse is on this rotating platform, actually zinc supplementation makes the mice makes them capable of better controlling their behavior in this rotarot test. And we also did this recognition task, which basically measures, like a proxy to a cognitive function of the mouse, how much time it would spend to investigate a novel object, how curious the mouse is. And what we see, so there is no difference between the genotypes, but the zinc supplementation improves this function or makes it better. So after that, my wife told me that I have to eat zinc on a regular basis, which I do. So what we want to do, together with Moritz Thiel and Anna Coy and Brewer, Benzeev and Galeheimer, we want to do a clinical trial on the, initially on the German cohort of patients to see what would be the outcome of daily supplementation of zinc in the dose which corresponds to the Wilson disease patients, to the GNAO patients, regardless of their exact mutation, for the period of 6 months. So this is what we want to do, but we are in the stage of looking for the funding for this project, which is ongoing. So basically, I am finishing up. So this is the type of analysis we do in the lab. We start from the patients with the exact mutation, and we try to understand, using all the techniques we have, what is wrong exactly to this mutant protein, which is then the basis for us to build the platform to screen. And we do screening campaigns actually for other mutations in addition to what we did before, because maybe we will find another approved drug which maybe could work for other mutations or all mutations. We are still hopeful. Once we do it, we have to validate it in the model systems with the idea that hopefully this can be fed back directly to the patients. So these are people in my lab which have been involved in these GNAO-related projects. And these are multiple clinical collaborators from different countries: Switzerland, Germany, France, Israel, Russia, Norway, Spain, Italy. I hope I haven't forgotten anybody. There are many of them, and I really appreciate their interest in building joint research projects and the funding bodies. And thanks for your attention.
Thank you very much, and I need to step up here because otherwise they shout at me. Well, we are really excited to hear this. This is major advances, and I think this is what all families would want to hear, you know, finally coming to a clinical trial, finally seeing hope that is getting closer, you know, to have a treatment or something. And it is really interesting to see that even in the animal that, you know, the model that you thought it was not responsive at all, eventually you saw some benefit from the treatment. So this is very interesting. Are there any questions here? One, please. Can you bring a mic?
I have a question, or actually a couple. So the first is, and maybe I missed it, at what human equivalence time point did you give the zinc or start the zinc? And then secondly, you mentioned that you know, you could use this model to predict how severe a given mutation would be. Do you have any thoughts on why we see such tremendous variability even within a given mutation, and what other factors might be playing a role in how that affects the studies on zinc?
So we add drinking water to the mice when they stop breastfeeding. Maybe we should also provide zinc to the mothers which do the breastfeeding, but this would complicate the interpretation. So regarding the correlation analysis, you have seen the graph. It shows sometimes quite strong error bars, so which reflects the fact that the clinical severity score which we use, even if it is a simple severity score, it shows strong variability for the same mutation across the patients. So which reflects the fact, which has been discussed multiple times today, that sometimes the same mutation produces quite a range of phenotypes in terms of their severity. Why this is so, I don't have a proper answer.
Please.
Thanks for your presentation. Really nice, Vladimir. I was just wondering, like, I think from C. elegans experiments, they've shown that RIG8 is upstream of GNAO1, at least that RIG knockdowns show the same effect as a GNAO knockdown. So now you show that it's a neomorph here and that it increases the binding to RIC8A and RIC8B. So I was just wondering about the new partners, the RIC8 partners that might explain the phenotype then in these patients. Do you have any clue about that?
Yeah. So you refer to studies, well, basically RIC8 proteins have been attributed to separate functions. They are partially interrelated, but they can be discussed separately. Initially, RIC8 was discovered as an activator of G-alpha subunits, a GPCR-independent GEF, guanine nucleotide exchange factor. Later on, it was discovered that it is a chaperone. And actually, there is basically not a clear consensus in the field, but some people would say that there is no GEF activity, it's just the chaperone. Others would say that the protein has two functions which are partially independent, but sometimes coupled. So in this regard, genetically, it can be upstream or downstream from G-alpha-O. So in C. elegans, there is just one RIC8, not two. In Drosophila, there is also just one RIC8, not two. In us, there are two RIC8s. One is responsible for the G-alpha-S subtype of protein, so RIC8B is there for G-alpha-S and G-alpha-OLF, and RIC8 is for basically all the others. But your question refers to the fact, is, if I understand it, what would be the outcome of the binding, massive binding of G-alpha-O pathogenic mutant to RIC8A and B. Actually, it's not just binding, it's also relocalization. The pathogenic mutants relocalize RIC8A and B from cytoplasm to Golgi. And we think, although we don't have clear data in favor of that yet, that actually we are dealing here with the universally important mechanism explaining not just GNAO-related disease, but a bunch of G-alpha-related diseases where the recruitment and trapping of RIC8s by the pathogenic mutants will affect dominantly the whole network of G-alpha GPCR signaling. And this aberration will result in neurological phenotypes for the protein which is expressed highly in the brain, as G-alpha-O does, but to other clinical manifestations for proteins which are present in other tissues. But at this stage, this is more of a speculation because...
So one question related to that.
We have little time, so
yeah,
very short question. We have also another one.
In the Golgi, it binds to KDLR and REP3A, I think, and maybe also to ARF1. So have you done pull downs to see whether there's more binding to this complex or not?
This is on our to-do list.
Okay, thanks.
Thank you.
Okay. Last short question. Thank you so much.
For this very, very interesting talk. I've got multiple questions, but I will just select two of them.
You can do some more in the coffee break.
Yeah. The first one is related to the nature of the mutation you studied in your work. Most of them were missense variants, so neomorphic mutation. Have you considered performing those tests on the either loss of function and mutation like the nonsense variant that were identified in GNAO1 related disorders or large deletion of the genes, and whether it will affect the zinc binding properties of the protein or the... And the second question is about the toxicity of the zinc, because in human pathology, especially with some disease, we know that exposing the patient to zinc for a long term can induce a decrease in copper plasma concentration and induce myelitis because it interacts with B12 vitamins metabolism. Have you noticed something like that in your mouse?
So no, but as I stressed, our tox study is preliminary, so we haven't done, for example, plasma measurements of the copper of these mice. So we don't see any obvious problems with the mice, but it has to be continued. But also stress that we use the extremely high doses in these experiments. And regarding the fact that, continuous zinc treatment may have some side effects, this is why we need the clinical trial. Of course, we want to do as much as possible with the animal models, but I think we are ready to start with the clinical trials. Regarding your question about the haploinsufficient mutations, so your work has, and work of others have shown that there is a separate category of the disease, basically, which is due to haploinsufficiency. When it is a deletion, it's not a missense mutation, it's not a new variant, it's not a neomorph, it's haploinsufficient. The question is, would some of the missense mutations also go in the same direction? So, for example, C215Y, which has been discussed many times, it is the weakest among the panel of mutants we studied in terms of binding to RICA. It's still higher significantly than the wild type. And for example, it binds to RICA-B not at all. I mean, it's zero binding to RICA-B. And we know that this is the mildest among at least the panel which I presented in the patients. So, and biochemically, it's also actually quite separate from the rest we studied. So does it mean that C215Y mutation is less a neomorph and more loss of function? I don't know. Maybe it goes in this direction.
Okay. Thank you very much. I think we had a great conclusion for this session. We all deserve a nice coffee now. Thank you. And we'll see you all exactly at 4:30 here, sitting in the room.